Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'll discuss about the DNA replication, mainly the initiation phase in eukaryotes. I'll discuss here. So this process, the DNA replication, this process is different in eukaryotes and prokaryotes, and the eukaryotes is much more complex compared to the prokaryotes. And this DNA replication, this process, it occurs in the before the cell division, and it occurs in the nucleus. So the initiation phase, it divides in two different phases: the pre-replication or pre-initiation phase and the replication or initiation phase. So pre-replication or pre-initiation phase, it takes place in the G1 phase. So in the G1 phase, the lot of proteins that required for the initiation, that all of these proteins are synthesized in this phase. So basically these all proteins are essential for the DNA replication. So this is why this G1 is known as also the preparation for DNA replication. Another phase is the replication or initiation phase that takes place in the S phase, and in this phase basically the initiation starts and that leads to the DNA replication, and the rest of the process occurs. So mainly the DNA replication occurs in the S phase. So this process, all of the process are very much complicated, so I'll just try to make it very simple. So let's have a look the steps of the pre-initiation and initiation phase. So as we know that in eukaryotes we have the origin of the replication in our genome. So there are multiple origin of the replication, whereas the only single origin of the replication in the prokaryote. So here is one complex, that is the ORC complex, that is the origin recognition complex, and this complex it comes and recognize the origin of replication site and it binds there. So upon binding of this complex of ORC complex, it recruits two different proteins complex, CDC6 and CDT1. So these all of the proteins name I already mentioned in my previous slide, so if you want to make sure you can check that. So mainly the CDC6 and CDT1, these proteins are known as the loading protein. These proteins are essential for origin licensing and limits the DNA replication only once in each cell cycle. Also the major role of the CDC6, also recruitment of the MCM proteins, that is known as the DNA helicase in the next step. So next step, that is the recruitment of the MCM proteins. This is not only the single proteins, this is the group of proteins around MCM2 to MCM7, so around 6 proteins are there. So upon binding, recruitment of these proteins, this is the DNA helicase. So mainly the role of this DNA helicase is to unwrap the DNA for DNA replication. So next step is the cyclin-dependent kinase activation. So upon activation of this cyclin-dependent kinase, these proteins, and then the CDC45 is recruited. So CDC45 when recruited, then the DNA polymerase alpha it's come and binds. So basically the recruitment of this DNA polymerase alpha, because of the CDC45 these proteins. So when this DNA polymerase alpha it bind there, then it starts the initiation process. So another points I want to mention here, that the CDK activation, and when the upregulation of the CDK proteins occurs, it starts the G1 to the S phase. So S phase starts from the activation of the CDK proteins. Alright. So next have a look. So here also I want to mention that all of the steps are take place in the G1 phase, but only from the recruitment of the DNA polymerase alpha, it starts from the S phase, DNA S phase. So all are the G1 phase, and this process only DNA polymerase alpha from this phase, this is from in the S phase, that is the initiation. So next is the initiation phase or DNA replication phase. So what happens here, So here, as you see in this figure, that this DNA fork, it's start here. So it means that this kind of unwrapping of this DNA, it occurs by the DNA helicase or MCM proteins. So after that, here what happens, the primer, so there is this kind of sequence. So this, you see, these two strands are separated, right? So then what happens, the primer, the RNA primer, it binds here. So this RNA primer is recruited by the primase enzyme. So here the RNA primer, it bind, and then DNA polymerase comes here, and DNA polymerase, it elongate this strand. So it keep on adding the nucleotide sequence one by one, and then it elongate this strand. So both of these strands are elongated by the DNA polymerase. But there are three different DNA polymerase actually. So first, initially the DNA alpha is recruited. So later on, the DNA polymerase epsilon and DNA polymerase delta, these two DNA polymerase also recruited, and these both DNA polymerase are responsible for the DNA replication in two different strands. Alright, so here, as you see in this figure, that DNA polymerase delta is here and DNA polymerase epsilon is here. So for both are responsible for two different strands. So this is about this DNA replication process. And also there are another two different proteins, that RFC and PCNA. These two proteins also recruited. So first, these RFC proteins are recruited, and then the PCNA proteins recruited. And the PCNA proteins function is to enhance the DNA replication, as well as it also, another important function is also there, that is for the DNA strand mispairing. Like if there is any mispair happens, that detected by this PCNA, these proteins. So this is about this strand. So another also one important point and I would like to mention here, that is the lagging strand and the leading strand. So what does that mean? So there are two, when these two strands are separated, so one strand is called the lagging strand and another strand is called leading strand. But the main difference of this leading strand and lagging strand is that the leading strand only a single primer needed. So like this is the leading strand, only single primer, this is the leading strand, yes, only single primer needed for the leading strand replication. And this is the continuous process of the DNA replication in this case. But in the lagging strand, what happens here, the lagging strand, there are also, it required multiple DNA primers, because it produces the short stretch of this kind of the DNA fragment. Like here, as you see here, this kind of short stretch, it's generated in the lagging strand, and later on these strands are joined by the DNA ligase. Alright, so this is all about the initiation process. So I think this is very much useful. So if you find this video is useful, so kindly hit the like button, share it and subscribe my channel. Thanks.